design more rational clinical trials in the future. And that's really where we're at. And we have to just keep picking away at it. So I hope I didn't upset anybody with that last little speech. Okay, so I've said this already. So again, when we talk about glomerular disease, when we give names like minimal change or membranous or the things you've heard today already, again, we're just describing what we see under the microscope. These are not necessarily specific underlying processes that result in these pictures under the microscope. And I made that point. And in the case of FSGS, we already know that this is true. We know it for a variety of reasons, some of which I'll talk about in a bit of detail. We know that a good subset of those patients who present with the microscopic picture of FSGS have underlying genetic abnormalities that result in it. A genetic abnormality, at least we know that there are familial diseases, abnormalities in genes that are passed on from generation to generation that produce this histologic picture of FSGS. I'll talk about that in more detail. People have brought up circulating factors. I think if you ask the average nephrologist, that nephrologist would tell you that 50% of patients who have the histologic picture of FSGS may have a circulating factor that causes this problem to occur. Where does that number come from? It comes from the fact that about 50% of patients who get transplanted get recurrent disease, often soon after they get transplanted. And so, as I think one of the people in the audience said earlier today, doesn't that suggest that there must be something extrinsic to the kidney that's causing this to happen, so there must be some circulating factor? Again, that's 50%. Can we remove it? What is that factor? We don't know. People are studying that, and you should know that. This has been looked at actually for probably 15, 20 years already. There are some new groups that are taking up the challenge, and I suspect some of them are actually quite excellent laboratories. I'm hopeful that they will be able to make some progress in this particular problem. It's probably one of the important holy grails in nephrology, and I really hope that we see an answer to that in the next several years. The technology is there to identify those types of things, and so I think it may happen. Now, in addition to things like genetic predisposition and circulating factors, there are a whole lot of other things that are associated with this picture of FSGS. There are things like if you take a rat and you take out one of its kidneys and 2/3 of the other kidney, and you leave 1/3 of one kidney left, if you just sew the rat up and let it go, that rat will develop FSGS in the remaining filters, the remaining glomeruli. And that concept, that observation, was made actually in the early 1980s and became a big area of investigation in nephrology. And by the end of the 1980s, we started using regularly ACE inhibitors and ARBs, drugs that you guys are all familiar with, because it was found that the use of those particular antihypertensive medications were useful in slowing down this process of scarring in the remaining glomeruli, in the remaining filters. And it's become now studied very carefully, clinically, huge trials, very reproducible. These are great drugs for these problems. But the point that I'm trying to make is that just the fact that remaining glomeruli go into hyperdrive, hyperfiltration we call it, may result in kind of progressive scarring that ultimately leads to the loss of whatever's left. So if you get hit with a primary process that causes, let's say, 60 or 70% of the filters to get knocked out, the remaining filters will take over the battle, but they may tire eventually, and the resulting picture is focal sclerosis. Okay. And that's true of some of these other things as well, and I'll talk about aging in a second. So you've heard about treatment. You've heard about nonspecific types of treatment like ACE inhibitors and angiotensin receptor blockers. these are very important treatments. They do make a difference. They should be used in all of your kids or in yourself if you're afflicted. I'm not going to go into any more detail on that today. I want to talk more about specific therapies. And when I introduce specific therapies to my medical students, I say, well, I don't know what the answer is. I'm almost afraid to teach you what we do because, truthfully, there's very little solid scientific data that supports what we do. I've heard it in this audience today, too. Isn't this an immune-mediated process? I don't know that it's an immune-mediated process, FSGS. Maybe a subset of patients who present with a histologic picture have an abnormal immune system that results in this. This concept of abnormal immunity or an abnormal immune system is the basis of our using all these toxic drugs, prednisone and Cytoxan and Cellcept and all that. We all use it out of desperation, I'll be honest. I use it with my own patients. Sometimes it works. Sometimes it doesn't. More often than not, it doesn't, actually, in my adult patients. But the point is that we are kind of still back in the dark ages a little bit, working from lack of knowledge but trying to treat in the here and now with our patients. So ultimately, what we'd really like to do is better understand in detail what the pathology is, what the underlying biology is that results in this abnormal histologic picture. And that's where the laboratory scientist is trying to make a contribution today. So again, we have limited understanding of the disease pathobiology. We have limited diagnostic options, and I'll talk about this a little bit. We can't define specific diseases necessarily, and we can't even predict which of our patients are going to respond to therapy. Wouldn't it be great if we knew up front that the child who presents with nephrotic syndrome is going to respond to steroids or won't respond to steroids before we start to poison our kids with prednisone? And actually, at least in part, we may be able to start to do that in the very near future. And as I've already said, because we don't understand the biology, we really have limited disease-specific therapeutic interventions available at this point. So I hope you don't go away discouraged with all this. The point is that a lot of research is needed. Your input, your pushing for it is what we need to get us to the point where we can make a difference. So things are beginning to change. And I have to say, it's kind of amazing for me to watch this. I've been kind of in the lab now for almost 20 years. And one of the things you see in science is that there are kind of waves of enthusiasm that occur in science. You know, there are a lot of people working in a lot of different areas, but something suddenly comes along that kind of really catches the scientists' fancy, and then everybody piles on to a problem. And that's when big progress is made in scientific research. And the really good news for this group is that that is really truly happening in the area of glomerular disease research today. I think the practicing nephrologist has always felt frustrated by our kind of lack of understanding. And with that as the initiative, doors are really opening scientifically that allow people to start to address these areas. And a lot of that has to do with the podocyte and podocyte biology in glomerular disease today. So why are we entering this new era? Because of big advances in genetics, primarily the tools of genetics that allow us to better understand or better investigate these problems, and then also new investigational tools of many other types that are allowing us to better study this really complex cell. The podocyte, many believe, is the center of the universe for the glomerulus, that most glomerular diseases, we believe, occur because of abnormalities or injury to the podocyte itself. Why is this true? Well, the first and most important concept is that podocytes, like neurons, generally do not replicate and therefore can't renew themselves. So if a podocyte is killed, or if it's sick and can't function, it does not have generally the capacity to renew itself. You know, if you think about it, your skin turns over every 3 days. Your gut lining turns over every 3 days. Somehow the podocyte is able to live an entire lifetime. And so it is susceptible, therefore, to problems because of that, I think. All of our cells are susceptible to injury of one sort or another over a many-year period. And in the case of the podocyte, if injury occurs, that can cause either podocyte death or loss from this basement membrane, this proteinaceous matrix that the cells sit down on. In general, if the podocyte is lost from the basement membrane, then what occurs is kind of a reactive scarring. And that scarring is what you guys think of as segmental sclerosis. Okay? So again, this is a key concept. I'm going to show you this again in detail. But if you lose your podocyte, you get scarring at that site. And enough podocytes lost is segmental sclerosis, the segmental sclerosis part of FSGS. Okay? So podocyte loss is a key kind of concept that is probably the common denominator of all forms of FSGS. Clinically, if the podocyte is injured, that's when you get proteinuria. And I'll show you this in more detail. If the podocyte is lost, again, clinically, you can actually find podocytes in the urine. They're actually, they fall off, make their way downstream through the tubule and out into the urine. And we can actually detect podocytes having fallen off the basement membrane today. And again, if scarring occurs because of podocyte loss, then one obtains decreased function of the kidney. You accumulate enough scarring, you lose enough filters, you lose enough nephrons, and you lose kidney function. Another important concept I've already made, and that is that not only is the podocyte the center of the universe for the glomerulus, but loss causes scarring. And there's good evidence for this. We know that this is true in humans in a variety of clinical studies that have been performed. And we know that this is true in animal models where we specifically injure podocytes. And I'll show you one example of that. So this is a model in which we can specifically target podocytes. We actually poison them with diphtheria toxin. And as a result, we can actually dial the amount of injury in and out. And so if we induce enough diphtheria toxin injury in the podocytes, we lose podocytes, and the result is that we get scarring. This is done in a rat, actually. So brown here is staining podocytes, and wherever you don't see brown, the podocytes are gone. They've been knocked off. And wherever there's no brown, there's also scarring. That's this pink stuff that's occurring here. So again, this is kind of direct evidence that if you kill podocytes, you get segmental scarring. Okay? And the more podocytes you lose, the more scarring you have in any one glomerulus. This is kind of a blown-up picture actually taken from a human, same type of thing. The browner podocytes that are remaining, the areas where the podocytes are gone, is where the scarring is taking place. And this can be very carefully quantified. So if one sits and counts podocytes and looks at the relationship between the number of podocytes and the amount of scarring, this PAS is scar, the more podocytes you lose, the less podocytes that are left, the more scarring can be detected in each glomerulus. So there's a it's incredible how linear the relationship is between podocyte loss and scar production.